drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today. Joining me.
Yeah, thank you for having me.
Do you want to just start off by telling everyone a little bit about who you are and what you study?
Absolutely. So yeah, my name is Nirosha Murugan. I'm an assistant professor here at Wilfrid Laurier University in Waterloo, Ontario, Canada. My lab predominantly focuses on understanding the biophysical dimension of life. We're trying to understand how optical, magnetic, and electric information is processed and integrated within the body to kind of help form shape and function, and how we can leverage those signals for detection of diseases.
And so when you say the body, are you talking about the human body?
Yeah, we're working primarily towards the human health side of things, but we do look at lower life forms in terms of slime mold, which we can probably get into, that don't have neural systems. So we kind of look at it across scales, but predominantly within the human system.
Yeah. And so on the one hand, we all know, very, very well from our lives that the physical world impinges on us. We see things and that is based on light, and we hear things and that's based on sound waves. But you said you look at optical inputs and you look at electrical and magnetic effects on humans. All of those things are affecting the human body?
Absolutely. I mean, on various scales and various dimensions, the electromagnetic spectrum itself encompasses light itself. So we're trying to understand really all of those, like you said, all of the physical dimension affects the body. The sun provides energy for us through, plants and foods and things like that. But there's also information within these physical signals that really what I'm trying to tease apart is like, we have these physical signals in our environment, but how are they interacting with our biology? And is there any information within them in which we can kind of tap into to see how that happens?
Mm-hmm. Yeah. And I think, maybe part of what we'll get into is how A lot of these physical signals, various forms of electromagnetic energy, can be detected and used by the body beyond the ones that are obvious to most of us. So if we start with, say, sunlight, for example, obviously we're visual creatures. There's a visible spectrum of light, but then there's an invisible spectrum of light that extends way beyond the visible. And we know, for example, obviously that UV light is important. We can't see UV, other organisms can, but it still affects us because, we can get sunburned and things like that. But, starting at a very high level, how does light, natural light from the sun, affect us and other animals beyond image formation? What are some of the major ways that light is regulating biology?
Excellent question. So we're all pretty familiar with the circadian aspects of the sun entrainment in terms of regulating a lot of our body rhythms. So when the sun comes up and goes down, we have circadian entrainment. So there's parts of our brain, the suprachiasmatic nucleus, that is tuned in to how much light that comes into our body. And when we think about human health, we kind of see that disturbances, for example, in shift working. Individuals who shift work who are kind of out of alignment from the sun's rhythm, they kind of have a lot of these pathologies related to heart, gastrointestinal issues that come about. So that's more on like the systems level, behavioral level scales. Now, if you scale down to the cell and tissue level, we see that a lot of the chemical energy that fuels our cells for mitochondrial function is all derived from light itself. So there's energy within the sunlight that gets propagated in terms of food that gets broken down to these cellular processes. Now, what's also exciting that we're starting to see is that like you mentioned, the non-visible spectrum of light, so the red to near infrared is also very important for a lot of our molecular synthesis. We see that, vitamin D, for example, we can't make vitamin D on our own without the help of external light. But also we're starting to see that wound healing, a lot of these molecular signaling function is all regulated by light. And we can kind of get into it a little bit more, but if we look into the molecular landscape of how receptors are distributed within our body, there are these receptors on all of our cells that are receptive to light. And they're in deep structures, for example, within our brain where we expect to see no light, deep structures within our body cavities that we expect to see no light. And that kind of begs the question, why are they there? If there are photoreceptors, are they involved in something more than just making vitamin D and then training these body rhythms? So
Mm-hmm.
that's kind of like what I think this movement is going towards, which is super exciting.
Yeah, yeah. So if I'm hearing you correctly, obviously we've got photoreceptors, we've got specialized structures in our eyeballs that allow us to detect light, and that part's obvious. Everyone knows that. And then the surface of our skin is light sensitive, right? The lightness and darkness of our skin changes in response to how much sunlight we get. Obviously, we can get sunburned, but what you're saying is like deeper within the body, the our cells seem to have quite or a lot of ability, a lot of detectors for light in places that we don't ordinarily think light is even getting.
Exactly. Yeah, so deep brain structure, they have these proteins called opsins, receptors. There's different kinds of opsins that are receptive to different wavelengths of light. So some of them are leaning towards the UV melanopsins that are within, again, deep brain structures that we see. So the fact that there are receptors, neuropsins, all of these different various kinds of variants of these opsin proteins, they are distributed heterogeneously within the body. So suggesting that there's some sort of evolutionary aspect of how we evolve to these wavelengths of light and how they're involved in terms of maybe internal communication.
Yeah, yeah. So we have opsins, we have light detector proteins literally in the middle of our brains. And so if I sort of map out the space of possibilities here, there's maybe two or three things to come to mind, and then I'll sort of let you direct us where the evidence
Sure.
is taking us. One reason we could have these light sensitive proteins deep in the middle of our brain is that, well, it's just a coincidence. They've evolved for some other reason and they happen to be light sensitive, but that's not really what they evolved for. Another reason could be that somehow light from the outside world is getting in there, even though it's deep within us. So maybe we've got long wavelengths of light that penetrate deeply in, and we are literally detecting with these opsins and things external light from the sun or from wherever that actually does get all the way inside of us. And then the third a possibility here that I think you'll be able to speak to is there could actually be light produced inside of us. It's endogenously produced. And so Is that roughly the space of possibilities there? Is there something I'm missing? And what do we know about what these options and things are doing deep inside of us?
Absolutely. I think the point two and point three, the fact that there's external light, different wavelengths, the issue of penetration has always been a topic of discussion in this space. But also internal light. So it kind of points towards, yes, we are evolved in this ball of fire, but what about what's literally happening inside of us? There's clear evidence that there's light being generated from inside of our body, inside of our cells. And perhaps those are why these receptors have evolved to detect and be involved in that communication. Um, yeah.
Okay, so light is being produced endogenously inside of us and other organisms. In other words, if we were to go into a truly dark room that had zero external light coming in, we would actually, with the appropriate sensors and detectors, we would actually be able to detect photons being produced from the cells of the organism itself.
Yes, and this has been well established now. We are pioneering this space, but this has been shown for over a century now with the
Hmm.
original Alexander Gurwitch experiments with onion roots, showing that, they didn't have the fancy detectors that we did, but they showed that if you have light impermeable materials like glass or quartz, you can kind of prevent cells from growing, onion root cells from growing. And there's no chemicals that are being emitted from onion roots. So it must be something physical. So the conclusion they came with that it must be UV light that's being emitted because of the barrier that they use to prevent it from getting from one side to the other. But beyond those original experiments, several people have shown using light detectors that these cells and tissues and us, humans, plants, all living biology emit light.
Hmm.
The big question that we are trying to solve as a community now is there information in that light? Can
Mhm.
we actually use that light emission to read what's happening internally?
Yeah, yeah. So it's unquestionably true that these, what do you call them? Endogenous photons, biophotons? How do you talk about this?
The terms have been thrown around over the last century, biophotons, ultra weak photon emissions. Now there's a movement towards autoluminescence. So
Hmm.
all of those just means unstimulated light. So this is not luminescence in the sense that it's not produced by a luminescent biochemical reaction. It's not light that's going in and being converted into another wavelength.
Yeah.
These are endogenous photons that are created from proteins changing, resonant frequencies, things like that.
Yeah, yeah. So it's truly endogenously produced. It doesn't depend on an external light source coming in. So these
Correct.
biophotons get made, and maybe they get made as a side effect of something else going on, and they're just sort of I don't want to say waste product, but they might not be doing anything. But as you've alluded, we might be really, they might be doing something functionally. There might be information there, and the systems may have evolved to not just emit, but detect these things in order to drive various aspects of biology. I'm sure we're going to go there, but let's start with basic mechanisms. What are some of the basic ways that these biophotons get produced in living things?
Right. And so that's been, again, another topic of discussion. So this entire space of biophotons has been pretty rocky until pretty recently.
Hmm.
And the reason for that are the tools that we need. In the genomic landscape, we've kind of made substantial strides in how we understand that side. With biophotons, the caveat that I want to kind of put out there in our discussion as early as possible is that we're limited based on our tools of what we can do and what we can understand, especially things of scale. So if we're thinking about where these photons come from, we have to think about the scale. Are they coming from organelles, proteins? Are they coming from subatomic units, electrons? For what we know as a consensus, what we can agree with is that it's tied to the energetic landscape of the cells. So if we look within the powerhouse of the cell, as we colloquially call them, the mitochondria, as the mitochondria function and they release these highly energetic molecules called reactive oxygen species, that is what we're associating the emission of light. So there's highly energetic states of electrons that get to a certain level. When they return back to ground state is when these photons get emitted.
Yeah.
So it's a continuous reaction of reactive oxygen species. There's some really good evidence now that's showing lipid peroxidation. So the breakdown of fats is also another source of these photons.
Hmm.
Ultimately, you're not going to get photons without changing ground level states of electronic values.
Yeah. So I guess that's basic physics stuff. And if I'm hearing you correctly, there's good reason to think, based on what we know about physical chemistry, that photons are going to get emitted as the metabolism of life is happening. But because it's probably very, very difficult technically to look deep, deep, deep inside a cell, it sounds like there's still a lot of fuzziness in terms of like exactly where all of these photons are coming from, the exact chemical reactions, the exact organelles, the exact places within cells.
Exactly. The body, the biology, it's messy, it's warm. So when we're thinking about physics, we have to think about all of these variables, temperature, the stochasticity of the system itself. So when things are emitted, there's always going to be, by things, I mean light is emitted, there's always going to be environmental absorption as well. So
Yeah.
what we detect from our detectors on the outside is going to be a subcomponent of what the biology is absorbing in its environment. And
Yeah.
water is a great one. Coherent water is another one that is involved in absorption of light.
Hmm. Yeah, so basically, biophotons can get produced and emitted, but by the time your detector as a scientist measures them, they've bounced around, they've been absorbed, they've been
Correct.
reflected through all of the milieu of the cell.
Correct. Yeah. And so that's kind of what we're trying to tease apart is, what is being absorbed? Is it being absorbed and then re-emitted? Is there a change in wavelength? Is there a change in frequencies? All of these domains that need to kind of really get addressed before what we actually pick up.
Hmm. And you mentioned water there, and you said coherent water, and I'm not even quite sure what that means. But what can you say about water in terms of what it might be doing in a biophysical sense? So obviously, water is necessary for life. We need to drink water. We know that there's all sorts of hydrolysis and chemical reactions that require water, and that's a big part of what water is used for. But is water doing something more biophysical and not just biochemical in our cells?
Great question. And let's think about that for a quick second. So you mentioned hydrolysis, and even within the mitochondria, creating the fact that you're creating energy produces water. It produces something fundamental to life. So when things are fundamental, they've evolved and to form structure. And what's really exciting on the physical nature of water itself, you have the production of protons, you have the production of these negative ions. So there's a distribution of charge. So on the physical landscape, it produces bioelectricity essentially across membranes and things like that. The other important things in terms of water, in terms of physics, is it's a thermal insulator. So it's going to absorb infrared light and help maintain the thermal landscape so that biology can function. So there's a reason why we function at 37.5 degrees as a unit. Water helps maintain that. I also mentioned infrared. So this is kind of like the speculative nature. So yes, thermal regulation, Brownian noise is maintained at that point, but is that also a source for us to absorb a certain amount of light that doesn't get propagated, bounced around to affect signaling? And that's sort of the speculative nature on the light dimension that water is associated with as well.
Hmm. Hmm. What there's a lot of different ways to go with this. Let's first talk more about the ultra weak photon emissions that you've worked on and that others have seen. So let's just define or redefine again for people, what are these ultra weak photon emissions, and what is, I don't know, you can talk about your research or just the field more broadly, but where's sort of the focus right now in terms of the wavelengths that are being measured by people like you, and where in the cells and in the body the action seems to be, if there's any special place.
Mhm. So just to kind of set the tone, ultra-weak photon emissions, UPEs, biophotons, they're usually within the visible wavelength, so about 280 nanometer UV all the way up to IR.
Mhm.
In terms of source, we kind of briefly mentioned mitochondria. We mentioned also lipid peroxidation. But there's a lot of individuals who are also showing that microtubules are also another important game players here in the biophoton community, in the sense that not only do they emit photons, but they also help propagate it. So the once you have light, they need to be structured and moved from place to place within the cell. And there's implication for consciousness, which you don't have to get into right now, that we can talk about later. In terms of photon emissions that are being actively studied within the human body, the first place that a lot of people point to is the brain.
Mhm.
Why? It's one of the most energetic organs within the body. So it's pretty useful to kind of modulate the energetic landscape and look at photon emissions. And people have looked at this from cells and the neurons in a dish all the way up to what we do within human subjects.
Mhm.
So also, I kind of want to mention some of the work that's been done to actually validate this work to show that we can do this within the visible spectrum is in plants. They've shown that if you tear a leaf, damage a leaf, wound it, that you do get these photonic bursts that are associated with reactive oxygen species. So it's a lot of correlative mechanisms that have been shown that if you induce damage that causes ROS, you do get photon emissions within the visible spectrum.
I see.
So that's kind of the current landscape right now that everyone is in consensus with.
I see. So biophotons get produced. They're often in the visible spectrum, but a variety of wavelengths can be produced. They are pretty clearly associated with, let's just call it heavy metabolic activity. So that's going to implicate the mitochondria, where a lot of metabolism is happening. That's why you've mentioned already a couple of times things like wound healing and damage. Anytime you need anabolic signals and more metabolism to build up structure to fix it, or I would presume to develop it in the first place, there seems to be more biophotons produced in those types of situations.
Mhm. Yeah, absolutely. And there's some fantastic work that's being done to actually show the specificity of these photon to cell types. If we are coming to the conclusion that it's metabolism based, we know that various cells are under different metabolic stressors and thresholds, and we're starting to see that those photon signals, those light signals, seem to match those energetic landscapes.
Mm-hmm. And so this is going to be a really fun conversation for me and hopefully the audience, because unlike most of my guests, um, for most of my guests, I have some reasonably good to very good background in the stuff we're talking about. So, so I have a lot to draw on. But with your stuff in the biophysics, I have much less of a background. So hopefully that means I'm going to ask naive but good questions. And it also means I have a lot more genuine questions. So, for example, you said something a moment ago. You
Mhm.
said you're talking about microtubules and you were talking about moving light around. I'm not even sure exactly what
Mhm.
that means. What do you mean by moving light around?
Great question. And um, first step is that light's being generated.
Mm-hmm.
If we're assuming that this light is not just spurious, it's not just background that's coming out of metabolic activities, it's involved in meaningful signaling, just like a gene that needs to get from the nuclei all the way to the Golgi apparatus to become a protein,
Yes.
there are these trafficking mechanisms that move these things, molecules around. And so if light is going to be involved in sort of meaningful communication, once it gets produced by the mitochondria, how does it get to where it needs to go? And there's the implication that the microtubule, the way that it's structured, the tubulin dimers are structured, is that they can propagate just like like these dynosins in microtubules, that they can potentially move and propagate that light from point A to point B.
Almost like a fiber optic
And this is
cable.
something that's correct. And I'm so happy you said that, because that's exactly kind of what we're working on in the lab, is that we're looking at aligned neurons, axons, and seeing how much of light, if we put light in, how much light comes out. And if it's like acting like a fiber optic cable, it should be 100% efficiency or near there. But if it's spurious and this is not really a propagation system, not like a fiber optic cable, we should get nothing.
Wow. So, I'm just going to say this now to remind us for later. Maybe we'll come back to this. But as someone who used to do a lot of optogenetics experiments where we're shooting blue light into the brain, when you read all those papers,
Mhm.
and I put myself back in those days, we always made the explicit assumption we don't have blue light sensitive stuff in our brain. And therefore, when we put in this light, it's only having our experimental effect. And therefore, it's not having some other effect we can't account for. Maybe that's not the case. We'll come back to that. So, let's just start very basic here. You're saying you can potentially actively traffic light around to serve a signaling purpose. So, by analogy, there's biochemical signaling systems in our cells. There's receptors, they're bound by other things. We have signal transduction pathways that take you from, say, the external part of the cell to the internal part of the cell to the nucleus. You might turn on a gene or start transcribing it more. That gene product then has to be actively shuttled somewhere else in the cell to make the protein. Like there's a lot of active regulation and shuttling around of biochemistry, let's
Mhm.
say. And you're saying that perhaps something similar is happening not with chemicals, not with molecules, but with light itself, that microtubules and other structures might be structured, they might be there in part to shuttle around light for information signaling purposes.
That's the assumption that we're trying to validate. And neuroanatomy and looking at microstructures of neurons kind of gave us the hint to kind of explore this route. So we look at the structure of a neuron, we see at the axon hillock, and you have the myelin sheath, and you have nodes of Ranvier where there is no myelin. And what's really interesting, if you actually look at those spaces, they have a high density of mitochondria in that space.
Hmm.
So typically when we look at neuroscience, we understand the electrical dynamics. Yes, that produces an action potential. That's great. And that's one source of information that gets passed down. So we've accepted that electrical information can be transformed into molecular and synaptic transmission. That's what we call that now. But if we look again at that node of Ranvier, we have mitochondria, and mitochondria produce light. So the assumption there is that the light that's being produced along those nodes are propagated perhaps through the microtubule along the myelin, that it perhaps is involved in facilitating some other level of communication, but this is all that needs to get validated. And kind of towards that area, the myelin itself is an insulative property electrically, but it also is an absorber of light as
Hmm.
well.
Hmm.
So there's sort of this dynamics between what's getting propagated and what's getting absorbed, but we need the tools again to kind of figure out at that scale what kinds of emissions that we're looking at.
Yeah, yeah. So there's some interesting possibilities here in terms of the information carrying function that light could
Mhm,
have. Like we're saying could, maybe. There's lots of stuff that needs to be validated. Let's just talk about the raw sort of biophysics of this. Can you speak a little
mhm.
bit about the molecular architecture of microtubules and what we know for sure about their ability to transduce light signals and move it around?
Absolutely. So the microtubules produced from dimers of tubulin themselves are highly polymerized, and they form this helical structure. So the helical structure itself maintains rigidity, the cytoskeletal integrity that we need to maintain cells. From a photonics point of view, they being tubulin dimers themselves, absorb within the UV wavelength.
Mhm.
So, which is really unique to any macromolecule that exists within the body, because as we know, UV is a high energy photon. So to be able to absorb without damage is a pretty unique nature to tubulin. So
Mhm.
that's what we know in terms of how tubulin is involved on the optics on the photonic side. And the helical nature is what helps propagate that photon.
I see.
Because those vibrational frequencies are match the UV wavelength, there's resonance within the microtubule domain that's within the megahertz to terahertz range.
Hmm.
So there's an internal resonance frequency or oscillation that's always happening within these microtubules that we're starting to understand is coupled to light.
I see. So everyone understands UV light's high energy light. It's very energetic. What you're saying, if I think about this as an evolutionary biologist, the microtubule dimers, the component pieces that make up the microtubules, they are capable of absorbing UV light. And UV light, as we said, is high energy. It, the fact that they're structured microtubules to absorb that light without being destroyed or damaged suggests that they evolved in the presence of a fair amount of UV light, even deep within the brain and body where UV light from the outside is not getting in. And so that would imply that
Mhm,
they evolved in the presence of UV light that was presumably produced quite locally to where these structures are located.
mhm. I mean, that would be a good, strong evidence for it. We also see this that when you depolymerize, so you break apart the structure, each individual tubulin monomer at that point is not able to move around light, obviously, because it's not structured, but the absorption also changes in terms of how much energy each individual monomer can absorb. So it's the full network that's involved in absorbing this high energy unit.
Mhm.
And it would be really interesting to see, how evolutionarily this would have happened. Though the sun has always been around in terms of our lifetime, the energetics of the sun in terms of the wavelengths and helio disturbances could also imply, evolutionary changes into how these microtubules and how these proteins have been made.
Mhm. So, you know, you gave us the raw biophysical fact that these tubulin dimers can absorb UV light. That's interesting, and perhaps we can talk more about that. I want to. How far does this fiber optic cable analogy go? Are there wavelengths of light that can sort of travel through the microtubules and get from one part of the cell to another? Has that been observed?
To my knowledge, not sure. These are experiments that we're actually ongoing right now. And we're using actually different species of spinal cords, as we know that different spinal cords from different species are aligned differently. Their microtubule and their alignment in terms of their axon distribution, their myelination is also different. This allows us to probe to the question exactly that you're asking. What's the wavelength that gets propagated? How far do they go? Is there a reason why we need to have nodes to continually produce light? So you get little bursts of light that get like a Morse code of light, let's say, in
Hmm.
terms of activity. But these are all things that we're ongoing right now in our lab.
Hmm. Hmm. So microtubules, when you learn about them in school, what they're most famous for is they're part of the cytoskeleton of the cell. They are physical structures that give cells their shape and their rigidity. They're important for like the cell cycle. So when a cell divides and pulls from one
Hmm.
cell into two cells, the microtubules are important cell skeleton structures that have that sort of physical spatial function to them. Are there any observations that we've made over the years in neurons or in any other cells where, the microtubules seem to be in the cell in a way that can't be explained by just serving as a skeleton for the cell? Is there anything that people like you have looked at them and said, okay, these microtubules seem to be in this part of the cell and they're sort of oriented in a way that we can't really just explain away with, they're a structural component of the cell or they're going to help the cell cycle or something like that?
That's a great question again. And what's exciting is that, much like the mitochondria that has been, coined as the powerhouse of the cell, and we're starting to see that it does more than that,
Mm-hmm.
the microtubule kind of got like a bad rap in the sense that it's just structural.
Yeah.
But it's definitely more than just structural. We see this also in magnetic stimulation. For example, when in researchers have shown that if you apply certain intensities of magnetic fields or electric fields, these microtubules move and they align. So the fact that it's not just that they are there for structure, just for support beams, they seem to be responsive to applied physical signals.
Hmm.
So that kind of then begs the question, are they moving and providing structure because of environmental signals? And they're kind of then altering cell signaling, or is it vice versa? So they are kind of like a physical antenna to propagate external physical signals, electrical, magnetic have been shown. And now we're trying to see, is it photonic as well?
Hmm. Hmm. And so, what do some of these experiments look like? And do we have the technical ability to really get firm answers to this stuff yet? And like, what do you think? What do you think sort of the landscape of discovery will plausibly look like the next few years in terms of nailing down what some of these biophotons might be functionally doing inside of cells?
Oh, I'm so glad that you asked that because, as people are listening, I want to think about and have them think about how we can build these innovative devices. When we've presented this work at many different quantum conferences where there's physicists and biologists, and there's always been a disconnect in terms of biologists, we know we need molecular tools so we can see and measure and quantify. The physicist asked the question, what's the scale and what's the setbacks you need? Like, what's the temperature setbacks? These light detectors, they're made for room temp, but when we stick it into an incubator, they fail. So
Hmm.
there's some of these innovative gaps that need to be made. So when we think about using these physical tools at biological temperatures and scales and CO2 and all of that is one gap that we really need to fill. In terms of, your question that you asked about how do we know, how do we visualize this? We're taking molecular tools that we understand in terms of imaging, for example. Imaging gives us a snapshot, and we're thinking about physics. A lot of the time scale gaps is something that we also need to fill. The photon, the speed of light, are we ever going to get a detector that can measure the speed of light? Not sure, but we can look at some harmonic away from it. So, I mean, that's something that I'm really passionate about and really want to fill is the tech development in this space to help measure the physical and quantum processes.
Yeah, yeah. I think what you're speaking to here is, for those who don't know, very, very often in science, there's this exciting and iterative dance that happens between discovery. So we're talking about this applies to any field, I guess, but in biology, right? You discover some stuff about biology, but then that also makes you realize, oh, we don't have the technical tools that allow us to really understand the next step. And then you have to have tech development, and that enables the next level of discovery. And then, this sort of process just repeats. It sounds like, compared to, say, molecular biology in the classic sense, this area of biophysics and biophotonics is at an earlier stage of this dance, and you're just starting to develop some of the tools that allow us to really start to measure things, but it's relatively early days.
Absolutely. And as biologists and scientists in general, we try to be creative and repurpose tools from other fields. We're using PMTs that are photomultiplier tubes and detectors that are used for telescopes to looking at the stars, microscopes to looking at, individual cell protein dynamics in terms of fluorescence. But there's still setbacks into those. And some new tools that are being developed right now to get to the idea of fiber optics. Are neurons fiber optic biological sensors? So there's some engineering development that needs to happen in this space. And with the way that biomedical engineering is going right now, it's a good time to make this movement happen a lot faster than it had in the past.
Mm-hmm. Can you give us a sense for some of the tools that have been or are being developed? What do they look like, and what exactly are they doing?
Absolutely. So for example, quantum dots, when people think of photons and quantum, they think of quantum dots, which are typically used to visualize biological cells or biological proteins, but they are working again at a stimulation. So you have to put in light to measure out light. One new development that's been happening at a company out of Quebec is interfacing fiber optic cables that are typically used in telecommunications, but interfacing them with biology. And to be able to stick a fiber optic cable inside of an axon, you need to create connectors that are biologically compatible.
Mm.
And so those are some biomedical engineering tools that are being developed to interface silicon-based stuff into wet,
Mm-hmm.
messy biology stuff.
So it sounds, if I'm hearing you right, people are developing literal physical fiber optics made in such a way that, say, if you put them into a biological tissue like a brain, they will be good at picking up the photons that are produced in that tissue. And so, in contrast to something like optogenetics, which is now widely used in neuroscience and other fields, you're not using the fiber optics to introduce light to do something. You're trying to just detect what's in there already.
Exactly. And like you said, optogenetics has come a long way from where it has been. And if we kind of go back to what you alluded to at the beginning,
Mm-hmm.
if you look at optogenetic data, and this is something that I noticed when in grad school, is that even the conditions that don't have the rhodopsins in them, the
Yeah, the control.
unstimulated, just the control conditions, there's an effect. And there's always been an effect, and
Yeah.
they're low. And usually, some people kind of throw out that data and say, "Oh, this is an artifact or something." The effects are low, obviously, compared to the channel rhodopsin conditions,
Yeah.
but that kind of begs the effect that the cells are still responsive to some level
Yeah.
of these
Yeah.
very intense lights.
Yeah. And let's
So,
talk about what some of those. Let's unpack this a little bit for people. So, um, what is optogenetics, and how does it work in the traditional channel rhodopsin blue light sense? Let's do that first, and then go to what some of these artifacts could be.
sure. Sure. So optogenetics is the use of light to modulate molecular landscapes, but it's involving the use of a very specific genetic sequence, channel rhodopsin, a protein that is responsive to light. So they couple it usually to another gene sequence that they want to upregulate or downregulate. And just like a switch, in the presence of light, those genes get expressed.
Mm-hmm.
So typically, that's what optogenetics is. Like you said, it's widely used from neuroscience all the way down to protein chemistry. So, um,
Yeah.
is that a good enough definition?
Yeah. So basically, over a decade ago now, some people realized, Carl Deiseroth and Ed Boyden and others, that there's organisms that naturally are sensitive to blue light and other wavelengths of light, like certain bacteria, and they have these special ion channels that are sensitive to blue light, and they open. So they basically said, well, let's stick these into neurons in a mammal and then stick a fiber optic into that animal's brain. And long story short, they figured out, and now people use all the time, you can stick blue light into the brain of an animal like a mouse that's expressing channel rhodopsin specific neurons and control those neurons. But now what I'm coming to with you is that the assumption there is that that blue light isn't doing anything except activating channel rhodopsin, to use this one example, because the presumption is that our brains are not naturally sensitive to these wavelengths of light. But what you said is actually, if you look at the control conditions in these experiments, you actually do see that something is happening that, may be small and is often dismissed, but it starts to get us to this idea of, well, is this blue light doing something even in the absence of channel rhodopsin?
Mm-hmm, mm-hmm. Yep. And I mean, that kind of leads really well into a field that's now becoming well established, which is the photobiomodulation community. So, photobiomodulation, those are using light without having a genetic construct inside of the biological tissue. So, it's just the application of light. With optogenetics, it's just quite interesting to see that these high energy lights are producing some effect within the neurons. In terms of what they mean or how it's getting propagated, it goes back to what we originally talked about, is we need the tools to kind of help understand how all of this happens.
Mhm. But just looking at the experiments at face value, like when you were a grad student, what did you notice? So in the control condition, when they don't have, say, channel rhodopsin, but they put in blue light, what do they see?
So in these very specific experiments, they were neural activity based. We
Mhm.
saw there's a change in membrane potential.
I see.
So change in membrane potential. There was also change in calcium influx. Though small, again, negligible. It's still not similar to background. So
Yeah,
there's some
yeah.
effect, but we don't know why or how.
Hmm, hmm. Okay, interesting. Um, and
And
so,
I just also want to, sorry, I just want to also point out, usually when these lights are stimulated, these light trains, the pattern in which these lights are stimulated, they're usually high frequency
mhm,
and they're sine waves. So they're just really repetitive oscillations that
yeah,
happen. What we're starting to know, and what we actually already know in neuroscience, a lot of the electrical firing that happens within the brain is pattern. For example, long-term potentiation that's typically associated with memory formation has a very specific burst firing of activity. And so when I was in grad school and saw this optogenetic effects with the sine waves, kind of brought us to the question, what if we patterned that light to match something that's biological? So we know that electrical activity has a certain pattern of firing. Why don't we match the application of what we put in to that firing? And we've done this work with blue light and red light, and we see that we can alter biological activity by just changing the pattern of light going inside. So kind of putting those two concepts together, that we see that in control conditions and optogenetics there's some change that happen, and if we take it a step further by adding pattern to what we put in, we can get more specificity in how we interact with those neurons, is again giving us a more of a story, a more maybe complete story, that neurons and biological tissue are receptive to light that's beyond just accidental noise.
mhm, mhm. And so if biophotons, there seems to be a relationship between biophoton production and metabolism. Brain tissue is a highly metabolically intensive type
Mm-hmm.
of tissue. There's lots of mitochondria. There's lots of biochemistry that's going on here that's probably producing these biophotons. One thing that I think you've worked on that seems like an interesting area of tech development is whether, and everything I just said sort of implies that there's going to be a systematic relationship between biophoton production and neural activity. And therefore,
Mm-hmm.
I would imagine you could develop technology that enabled you to detect biophotons as a readout of neural activity. What's going on in that space?
So yeah, you alluded to it, and we published this paper not too long ago, from human subjects, that we have developed a tool to pick up brain photons from living subjects, and try to couple them to their cognitive state. So much like the development and evolution of electroencephalography, so looking at the electrical signals that are coming out from the brain, we try to stimulate or simulate that development using light. So we had developed a system where we had multiple sensors, photon detector sensors, that are placed around the head, at very strategic places based on the task that we wanted the participant to be engaged in, that we expected to be more activity, and looked at what is coming out at various times in their process.
And how do you
And
actually
that was the
do
question that we wanted to answer.
Yeah, how do you actually do that? Specifically, what are you detecting here in terms of the biophotons? Like, how sensitive is the signal? What exactly are you measuring coming out?
Great question. So the first big hurdle is how do we do this in an environment where we're covered with light?
Hmm.
How do we pick up these very, very weak signals? And to give you sort of like an analogy, we are picking up about 10 to 100 photons per second, which is 1,000 times weaker than the moonlight on a very dim moonlight day.
Yeah,
So it's
yeah.
very, very, very subtle, very, very weak. And so we have developed a what we call a hyperdark chamber, which is a room that is very, very dark. It's a room within a room, no lights inside of this room. And all of our grad students are pretty dark adapted now in this space. Along with this, the room is also lined with with copper, so it's also a giant Faraday cage. So we shield this room from magnetic fields, hyper dark chamber, and these detectors are placed strategically again over the head, right up against the head, and
Yep.
we record signals. We turn the devices on, and we get the participants to engage in a task, and we're looking at the photon emissions over about an hour period.
Okay, so it's literally a photon detector in a very, very, very dark room that's also sort of insulated against electromagnetic stuff, generally speaking. It's in a Faraday cage. It's a room within a room. It's totally dark. Before we even like just talk about the technology and the science here, what's it like going into that room? Have you sat in there for any length of time?
I have. We spent a lot of time in the building that room and validating that room because there's things like heat that gets picked up in there that we've had to make sure that it's not getting picked up by our sensors. It's actually quite calming. It's very meditative. It kind of gives us this sense of how much light that we're really exposed to on a regular basis, that when you go into something that's super hyper dark, you're very aware that it's very dark.
Yeah, yeah.
So that
It's
kind of
much
brings
darker
another
than
dynamic.
even it's much darker than even a what we normally think of as a lightless room.
Oh, absolutely. The dark curtains that you probably use in your bedroom is not dark enough. So there's still light everywhere being generated.
Yeah, yeah. What can you give me a sense for like how dark is it in there? Is it truly like zero external light bouncing around?
I mean, we have quantitative measures. Our background values are around 1 to 2 photons per second. We can tell when the light gets turned on in another floor right above us.
Wow.
We get about 1,000 photons per second. So kind of gives you a very, maybe an analogy of how
Yeah.
dark it is in this room.
Yeah, again, maybe a simple question because I'm not a physicist. So if someone turns on a light in another room and you get like 1,000 photons in your dark room within a room, how
Chamber.
does that light get
Yeah.
in there?
Light can travel through things.
Yeah.
There are very few materials that is impermissive to light. There's vents. There are, buildings are not so light tight like that
Yeah.
that,
The
that we
light
know.
bounces around and
So
literally goes through.
photons will find a way.
Wow. Wow. Interesting. Okay, so you are literally detecting. So you put people into this super dark room that you've specifically engineered to have all of these special features. It's very well insulated against light, but it's not perfect. But you can put a photodetector of some kind against someone's skull, and you're emitting photons that
Mhm.
come from their brain through the skull and out.
So that's what we're measuring. That was the original hypothesis, and this is built off of almost a decade's worth of work of people who've shown in culture that if you alter neural activity, let's say with adding glutamate or any other neurotransmitters, you are getting different signals coming out of neurons in a dish. You've scaled up. There are studies that have shown that if you put light detectors in a cranial window of mice and rodents, that there are photons again coming out. So, in all of those studies, it was brains or brain cells, but we wanted to scale this up to a whole human with just light coming out of their skull.
Mm-hmm.
And from what we see and what we have shown, we can.
Mm-hmm. So, okay, so you can develop this type of technology, whether or not these biophotons have any biological function, whether or not they evolve to as a signaling, a way to do signaling inside of brain tissue, even if they're totally passive, they still will have a systematic relationship to metabolism and to neural activity. And so, no matter what, we should be able to develop these types of technologies to use light detection as a means to ascertain something about what brain tissue is doing. But on the question, the unknown question of whether these biophotons are really serving specific functions, what are the major hypotheses there? What are your intuitions, and sort of what experimentally, where are you guys looking in terms of where you think it's most likely that we will find such a function for these biophotons if it does exist?
Excellent question. And this kind of helps us think about really the role of light in
Mm-hmm.
biology, in human biology. The first question is what's coming out? How much is coming out, and can we detect it? That was the very first question that we got, and that we came up with. And in the paper, and what others have shown is that compared to background, which is what we call, thermal radiation, the amount of light coming out of the brain and out of the skull seems to be space dependent. So we know that if we look at the skull bone anatomy, the part of our skull that's against our temporal area is much thinner than our frontal or occipital. So the amount of photons that we get should scale with that, and
Hmm.
it does.
Yeah.
So just from the skull anatomy itself, we're starting to see that it's not just random, it seems to be specific to that. And then there are other questions and other hypotheses that come up. What if it's just coming from our skin? What if it's coming from the vasculature on our scalp and not
Yeah.
actually from our brain?
Mm-hmm.
That was always in the back of our minds. And depending on the task that you do, the reaction rates and the timing differences doesn't match what vasculature would show us. So the hypothesis there then it must be coming from electrical activity. Now we are trying to kind of get to this question about melanation, different pigments absorb different wavelengths of light. Do we see that also in our study? We've had various kinds of participants with hair, no hair, colored hair,
Mm-hmm.
different types of hair. And we do see a melanation dependent response. So again, it's sort of like building on the story, but every time we try to answer one, 10 more come up, and I guess that's the joy of science. But the next hypothesis that we're trying to test is the propagation. Is the light coming out just random? What our first paper showed that it's not random, but how is it getting there? If it is
Mm-hmm.
generated from neurons, how is it getting from the neuron to our detector?
Mm-hmm.
And what's being lost?
Mm-hmm.
And those are what we call the nerve guide experiments, what we talked about at the beginning.
Yeah, yeah. So you're sort of narrowing in on what could become something very interesting here. So you said you've determined or you've ruled out that the light coming out is totally random, that it's just sort of emitted as a byproduct of something. I guess there's assumptions here, like if it's totally random, it's going to scatter in a particular way through the tissue, et cetera, et cetera. How did you exactly rule out that it's non-random in terms of how it's being emitted? And yeah, how do you think about that?
We can't completely rule out that it's fully, I guess let's stepping back a little bit. The randomness aspect came from our, or teasing out whether it's random or not, came up from our experimental design. So I kind of want to kind of explain that a little bit
Yeah.
more. So doing a lot of these kind of experiments, we had to be very specific on our task. We can't do anything visual because you can't see anything in those spaces. So we picked a very auditory task. So where in the EEG or the electrical landscape, you should see changes within the frequency bands associated with auditory processing
Yeah.
within the temporal lobe. So we picked a task that was very quick and we should see changes. And we validated this. That's why we did simultaneous experiments with EEG and the light emission. Because originally my hypothesis was that it would track. When we see spikes in electrical activity, we should see spikes in photonic activity because if we're saying they're electrically related. And that was an assumption that was proven false, that they don't track one to one. And so that result tells us that there might be some sort of scattering happening. So, and there might be some sort of
Mhm.
loss of photons that is happening. Because
Yeah.
if it was truly one to one, every time we got an electrical spike or increase in frequency in those spaces, we should get more photons, which we don't.
Mhm. So basically, you're saying if biology has evolved such that light is being actively moved around in certain patterns and it's being used for something functional, we would expect it to scatter through the tissue in a different pattern compared to if it was just sort of emitted and this was all just sort of a passive thing. And you're seeing
Correct.
hints that that is the case.
That's exactly what's happening. And the current experiments right now, which I can kind of allude to, is we're using wavelengths of light. So if it was truly random, we should not get wavelength specificity in what is coming out. But in fact, we are. We're getting very specific wavelengths that are tuned to the emission in terms of the tasks that the individuals are engaged in.
Hmm.
So that is also another indication that these are not random. They're tied to some form of activity. Um, and the other question that we're trying to also answer is where are they coming from? Are they deep brain photons that are bouncing around and making their way out? Are they cortical? These are, again, we'll need some more software to kind of help source localize where those photons are coming from.
Yeah. And so, I mean, when I started to read about your work in this general area, it made me think of a different period in scientific history, which is very famous, at least for certain types of scientists. So when you go back to what people would often call the early days or the the days that preempted the molecular revolution and discovery of the genetic code and stuff, you actually had this period of time where a lot of physicists started getting into biology for the first time, and that essentially is what gave birth to molecular biology. It was basically physicists coming into biology. And there's a very famous book from that era. I think it's a book or an essay by Schrodinger, who's famous for being a
Mhm.
quantum physicist. He's basically a math guy, and he had an essay or a book called "What Is Life," and he thought
Mm.
about the question of what biology is from a physics perspective as opposed to just a biologist perspective. Can you talk a little bit about some of the concepts he brought in there and how you start to think about this question of what is a living organism in a physics sense as opposed to just a biochemistry sense?
Wow, what a profound question. I'll attempt to answer it. What Schrodinger discusses in What Is Life, and I think what the movement that is happening right now is looking at first principles. First principles that define physics, the formation of energy, the transformation of energy, and the utilization of energy in a structured format. And so what I'm really trying to engage in, and I think what he tried to express in that essay was that energy needs to get transformed and structured in a meaningful way. And that meaning draws from material properties of what that biology is.
Mm-hmm.
So we're all going to be, microtubules or plasma membrane or cell membrane lipid dynamics. So there's aspects of constraint of fundamental principles, energy driven, that's going to determine what biology can do. So there's one dimension of constraint and structure, but then the other aspect of life is information. How do we understand how information gets transduced and processed? And you mentioned the molecular revolution that happened. And what we think of information right now is chemistry, is genes, molecules, proteins, which is one dimension, but we are completely missing the physical. How does the physical landscape process information in terms of these biological constraints? So kind of tying in what Schrodinger had mentioned in terms of information and energy, I mean, that is what I'm trying to solve right now in the work that we're doing.
Mm-hmm. So one of the things that you said on your social media that ties into this stuff is you said that biological structures behave as metamaterials, constraining electromagnetic, mechanical, and quantum fields, shaping how energy propagates, resonates, organizes everything from molecules to minds. So basically, what we're talking about here is if when we point a telescope out into the middle of the universe, energy is relatively unconstrained. Things are just moving around as they do according to physical principles, but life in some sense seems to be a structure of matter that changes or patterns the way that energy flows in a very, very different way. So what exactly were you talking about there? What is a metamaterial and how do these things tie together?
Great. A question that I really want to dig into. So a metamaterial is materials in which pattern information, they have intrinsic properties of that matter itself that's going to guide how it forms and how it interacts with the environment. For example, refractive indexes for light. The brain itself can be viewed as a metamaterial, like the fats, the proteins that are within it any magnetic stimuli, for example, in transcranial magnetic stimulation, the fact that the brain is a metamaterial will allow for applied information to get altered in some way. We also see this in terms of permissivity, patterning of how certain materials get structured together. So it's just intrinsic patterning of that material because of the way that the atoms are organized
Hmm.
that will give rise to how it interacts with the environment.
Okay, interesting. And there's something here that has to do with how biological systems pattern energy flow through themselves.
Hmm. Absolutely. So, like, if you take, let's take the myelin. The myelin, why is it like a jelly roll? Why is it not just a big sausage of fat that's across the
Yeah,
axon?
yeah.
The that aspect, the fact that it's a roll, produces a dielectric current, um, physically. And is that involved in the patterning of how electrical impulses pass through the axon? To be determined. But there's something unique of the fact that the myelin sheath produces this property that we need to understand how it is involved with neural communication.
Hmm. So another question I want to ask you, that's it's sort of a big question and it's vague, but that's at least half on purpose. So I want to ask you the question, what is energy? And before I let you take the reins here, this question has, sort of different angles you can take on it. Well, in one sense, Einstein's equations tell us energy is mass times the speed of light squared. So physical stuff has a mass and it's bound up with this really big number, the speed of light, and energy is that. From a biochemist's perspective, when we think about energetic processes in life and life, ATP and mitochondria, and the energy currency of life, energy is sort of about the stability and instability of chemical bonds and how those things are moved around. You're a biophysicist, but you're in also the realm of metabolism and biochemistry. How do you think about this question of what exactly is energy as it relates to biology?
I'm smiling because I've had this exact debate very recently. And from a physics point of view, we understand what that equation means. But if we try to really understand what is energy across scales, from physics to biology,
Mm-hmm.
it's really the potential for change. It's potential for some kind of movement. If we take a water molecule between the hydrogen, between the oxygen, that bond is the potential energy. Only when it breaks, when you have that change, do you get the formation of that energy will be used for something else. So that bond is the potential for energy. So what I think of what energy is potential. It's the ability to have some sort of change in a system. And we call that kinetic, chemical, electrical, potential, whatever. But fundamentally, it's the potential that we have for change in any unit that we look at.
Hmm. Hmm. And so, okay, so it's the potential for change. That's pretty intuitive. The chemical bonds correspond to that potential in the context of the biochemistry of life. And, this is where we start to reach the limits of my knowledge here because I'm not a physical chemist and I only vaguely remember things about molecular orbital theory. But those bonds, in some sense, are the way that electrons are orbiting around the atoms and how stable they are.
Yeah, I mean, then we kind of go down to the quantum realm
Yeah.
and
Yeah.
subatomic realm. Then we think about probabilistic nature of where electrons are going to be within their valence shells, but it's still the potential for those electrons to interact with another set of valence shells within another electron. So the definition of potential for change still holds true. It's the uh, the intrinsic ability for one unit or system to be able to interact with another system, and there's going to be some sort of constraints that allow for that transfer of change.
Hmm. And so when we think about chemical bonds breaking and forming, whether it's in the context of ATP synthesis or energy use in a mitochondria or any other aspect of biology, bonds get formed, they get broken. That's tied to this concept of energy and the potential to do work and move things around. And those bonds are very much they are the electrons doing different things. And I think this is also where we start to talk about like the basic biophysics of the light production, right? Because isn't the light
Mhm.
produced based on what the electrons are doing in terms of going back to their ground state and moving around and things
Ground
like that?
state. Exactly. So that kind of brings me to this new way of thinking of how physics really interacts with biology. you know, going through the molecular education system, as I call it, we are always taught that there's a receptor and a ligand, and you have some sort of resonance vibrational, and that produces a signal transduction. And kind of moving into the physical domain, we are always thought of looking for what's the receptor that's going to interact with light? What's the receptor that's going to interact with magnetism? And I think we've been thinking about this wrong. I don't think we should be looking at receptor physics interactions. I think we should be looking at the energetic landscapes and how those thresholds change. So if we think that a cell or even a protein has an energetic shell and we apply more energy to it, are we then rate increasing or decreasing that energetic capability? And then does that get transduced? So if we kind of again reframe our understanding of what energy is, potential for change, and I add more energy in the form of light, am I increasing that potential for change? And we can call that bonds breaking. And does that then release an ROS or not? So I think there needs to be a little bit of shift in how we think what energy is and how energy interacts with applied energy.
Yeah, yeah. And there's so much sort of subtle and implied and potentially disturbing richness in some of the things that you're saying or what's implied by the things that you're saying, because I'm going totally off the cuff here, but when we think about the extent to which electromagnetic energy, whether it's light or in some other form, affects biology, to the extent that some of the things you're articulating or some of the things that are sort of implied by what you're articulating are true, this would start to bring us to things like, okay, well, all of the experimental conditions under which virtually all of biology has already been done has been in a restricted and unnatural electromagnetic environment. And so what is that going to mean if that is affecting the types of results systematically across all experiments that we see in biology up till now?
I mean, there's some really cool experiments that have been done or observations that have been made. For example, in stem cell research, they've shown that individuals who take their plates from the incubator and they put them on their desk and they put them on these carts that move, that the vibration itself can induce the differentiation in a certain format. And as molecular biologists, we try really hard to reduce variance, to reduce variability between our conditions and try to keep them, homogeneous. But, for example, the fact that mechanical vibration can change stem cell fate, we also see that applied electromagnetic stimulation can change stem cell fate, says that sensitive biological states are responsive to physical signals. And another interesting observation that was recently published is that within an incubator that every molecular biologist pretty much uses, there's gradients of electromagnetic fields
Hmm.
within the incubator.
Hmm.
And so the heterogeneity that you might get between labs or even between people doing them on different shelves
Yeah,
can
yeah,
maybe induce this variability that we didn't know where it was coming from, but it actually could be coming from the physical dimension that is transduced to the molecular.
yeah.
But again, the more we understand of how that happens, will kind of give us that complete picture.
Yeah, yeah. Yeah, I mean, basically what you just said is, yeah, a lot of the stuff that we think of as noise and random experimental variability in the results we see, it's not, I guess, true. I mean, it is noise, but like it's actually coming from somewhere that's potentially has a very simple explanation that we just didn't consider. The petri dish in the back of the incubator might behave a little different than the one in the front because it's actually exposed to a different electromagnetic environment.
Correct. And I kind of want to go back. What is noise? Like, what is noise and
Yeah,
what is
yeah,
information? Like, why are we defining it? What is, so I think there's like a semantic gap here of what
yeah.
is noise and what is information. But again, I think that's something that needs to get worked out.
Yeah, I think it was. I'm pretty sure it was my PhD mentor. Actually, I did systems neuroscience, and there's this concept of spontaneous as opposed to evoked activity in neurons, and we kind of
Mhm.
think of the spontaneous activity as being noise, or at least partly noise. But I think he said something like, "Well, spontaneous activity is just activity we don't understand yet. We don't understand what's evoking
Yeah.
it."
Yeah. So noise is just a bucket that we don't really get,
Yeah,
and information is maybe something that we understand and patterned.
yeah, yeah. So how? I mean, given what you study and the things we've talked about already, how are you, like, I'm going to try and tie this back to things that ordinary people think about and sometimes get freaked out about. Are you like, what Are you concerned at all about 5G and cell phones and all of the signals that we assume or hope are relatively innocuous that our technology depends on? Is there more to that than meets the eye, maybe?
I'm not worried. And I understand why people are worried because, it's a scary thing that we don't understand. And when we don't understand something, that typically produces an anxious state.
Mm-hmm.
I'm not worried because as someone who's trying to understand the physical dimensions of interacting with biology, the in my view, the biggest key to all of this is information patterning. If I blast, if you take the chemical domain, if I just dump a huge high concentration of anything into a system, even the most innocuous thing, like let's say coffee, it's going to kill us. It's going to kill the system. High enough concentration of anything will damage biology.
Mm-hmm.
And that's kind of where the electromagnetic scare comes in. When you put high enough intensity of anything, it'll damage the biology. With magnetic fields, though there's a lot of heterogeneity in the literature that's being produced in this space, pattern is the most important. You'll see that, at the right frequency, at the right intensity, the right makeup of those variables, then can you interact with biology. And that's really something that I'm trying to validate using conventional molecular tools to show that the information within these physical signals of whatever parameters, it's what ties to unlocking the biology.
Mm-hmm. Mm-hmm.
So I'm not too worried about it. There might be some, hazardous effects that we may not know about, but that's only going to come about until we test them.
Yeah, yeah. So as you've mentioned multiple times, we're sort of at the frontier of this world of biophotons and what they're doing. We've just started recently, people like you, to be able to measure these things in order to study them and pursue some of these questions. But you've mentioned that you see a lot of these biophotons when a lot of metabolic activity is happening. You see them, at the mitochondria. And you mentioned briefly, I think at the beginning, when tissues are damaged and they regrow, obviously there's a lot of metabolic activity that happens at those locations at those times. And this brings us to the field of regeneration. I believe you're doing work in this field. What are you guys working on today? And give us a concept of what are the major model systems in regeneration that are famous, like some of the salamanders and stuff like that?
Sure. Yeah. So salamanders are well known with regeneration. Axolotls, they're very cute, but they also have this really cool ability to regenerate parts of their body. But they have innate stem cells to allow for them to repopulate what they lose. Unfortunately, we don't have that luxury. We don't have a huge population of stem cells. So in tissue engineering, what we're trying to do is use latent developmental pathways that were once regenerative. Like when we're in embryonic state, we're pretty regenerative. And to kind of kickstart those mechanisms. So in my postdoc, I worked with Michael Levin and David Kaplan, and we developed a silk hydrogel delivery system to kickstart latent developmental pathways by delivering multiple compounds, which was like a novel discovery. So we delivered 5 different compounds to help kickstart some signaling inside of those tissues to repattern them into a fully functional leg. So that's where our model system was in limb amputations in xenopus frogs that are not regenerative in adult states.
I see.
And there was a reason why we picked frogs.
Yeah.
Sorry, go ahead.
Oh no, I was just gonna say, okay, so there's organisms like salamanders and other things. You can cut off their arm, and their arm will regrow because they've got these stem cells that they retain even in adulthood. There's other relatively closely related organisms like these frogs that I think you're talking about, other amphibians. They don't have that regenerative capacity, but you are able to induce a regenerative capacity in those creatures, even though they don't normally have it.
Correct, correct. And I also want the caveat in being that when they are young in their tadpoles
Mm-hmm.
phases, they are able to be regenerative.
Mm-hmm.
But as they go through their developmental period and metamorphosis, they lose that ability.
Yep.
And there was a reason we picked this model system because much like us, when we are in utero and in embryonic stages, we have pretty regenerative capabilities. But when we're born and when we get older, we lose them. Specifically, for example, in digit tips, if up until the age of 4, if a child loses the tip of their fingers, they're regenerative. They
Hmm.
can able to kind of grow that back. But if we were to cut it off right now, nothing, we'd
Mm-hmm.
have
Mm-hmm.
damaged tissue. So it was a good model system to try to understand how do we kickstart developmental pathways.
Mm-hmm. And so it sounds like across animals, there's greater regenerative capacity as you go earlier in development, because I mean that's what the early embryo is. It's generating the whole what will become the organism. As development proceeds, the organism tends to lose regenerative potential, but in some creatures, for some reasons, they retain quite a bit of regenerative potential even into adulthood, and in other ones there's very little. But even in human beings, up till about the age of 4, we have some regenerative capacity in our fingertips. And so the work you did in frogs, it ties into this idea that, okay, if regenerative capacity goes down across developmental time, perhaps there are ways we can reawaken it even in the adult animal.
Exactly, exactly. And brings us some sort of evolutionary questions as well. It's like, why did we lose them?
Yeah.
What was the cost? Was there an energetic, metabolic cost to losing them so that we can gain it elsewhere? So these are some interesting questions in regenerative medicine that
Mm-hmm.
should be asked and
Mm-hmm.
answered.
And how did you reawaken this regenerative potential in the Xenopus frog? You said you added a few compounds. Were these small molecules? Were these transcription factors? What was the biology there?
Mhm. So our main goal was to, and much like with many of my research projects, is to not micromanage from bottom up, so not from genetics up, but to from systems down kind of control. So that we had to solve this in two problems. One, can we create an environment that will even allow for regeneration? And then two, what are we putting into that system to kickstart those pathways? So the first dimension of microenvironment is using silk hydrogel. There's a reason why we picked silk hydrogels, because on the physics side, we see that if you increase stiffness and you change the stiffness components interacting with wound healing, it'll induce fibrosis or scarring. So the mechanical landscape is very important. And so that's why we used silk hydrogel. So the water is also very important in wound healing. In terms of the chemical compounds, we picked 5 very specific compounds, hormones and proteins that target very specific pathways in wound healing that prevent scar formation and induce, like, for example, we had brain-derived neurotrophic factors, BDNF, well-known compound associated with neural plasticity, neural growth hormone, for example, that targets the metabolism and bone formation. So there's each of those compounds were selected based on their individual capabilities. And using our device that was silk based, we delivered it to the wound site after a full limb amputation in frogs. We applied it for only 24 hours, removed the device, and let the animal take over from there.
Mm-hmm. So it sounds like so there's a biochemical component to this. You introduce certain factors that are like growth factors that are thought to induce or known to induce, the building up of tissue, but there's also a purely physical component to this. The actual physical material that you used was important for how the regrowth happened and whether or not you had more or less scarring.
Absolutely, absolutely.
Wow.
And this is now becoming a well-known domain on the physics side, is that the mechanosensitivity or mechanotransduction, there's a interplay between the stiffness, the mechanics of things, and how information gets transduced to the chemical landscape. And there are, for example, piezo channels, mechanotransductive ion channels, for example, that we know that are on the skin, but we're now starting to see that they're also within tissues as well,
Hmm.
within deep tissues.
Hmm. And then I think earlier you also mentioned that wound healing is affected by light as well. What do we know there?
Absolutely. Absolutely. Again, going back into the photobiomodulation community, near infrared and red light is now well established for anti-inflammatory properties and for accelerating wound healing. Red light has also been implicated in angiogenesis, so increasing more vasculature to promote wound healing. So if you have more vasculature, you have more nutrients getting to the site to help heal and close the wounds. Again, how is the missing dimension?
Mm-hmm.
What is it about putting light into what is, I guess if we rephrase, what is it about putting energy into the system that allows biology to interact with it
Yeah,
to
yeah.
change the chemical landscape?
Yes, we know that wavelengths of light in the on the red side can affect these things, but we don't know exactly how that's happening. But at some level, it must probably be, the mitochondria are sensitive to those wavelengths. Obviously, they're involved very intimately in what the cell is doing from a metabolic perspective and an energy use perspective. So probably there's some kind of mitochondrial modulation happening that is influencing how these like anabolic processes actually play out.
Absolutely. I mean, even to this point, we know that cytochrome C oxidase and complex 4, these important proteins within the mitochondria, have wavelength specificities. They seem to be red leaning. So it could make sense that that's why red light seems to be working for mitochondrial function. But there's a new perspective that I've been working with Martin Picard out of Columbia with, trying to understand the energetic resistance, so that
Mm.
certain systems are at certain thresholds of energy levels. And based on their molecular landscape, perhaps we can alter these resistance levels to incoming energy inputs, for example, light magnetic fields. Um, Again, theories that need to be validated, but it kind of helps understand from a first principles point of view. You asked the question, energy. What does that mean? How is it getting manipulated in these systems? And what does light have to do with it, as a form of energy?
Yeah. So in the context of regeneration and wound healing, which, I suppose these are just, points along a spectrum. Regeneration is just sort of a very impressive form of wound healing, it seems. But, we have some capacity to restore our tissues when we're injured. And you've told us now that physical aspects of the environment, like light and light, just the tension and the rigidity of the mechanical stuff that our bodies interact with, affects how these regenerative or healing processes happen. One area this immediately makes me think about is, wow, every time we get a cut or a broken bone or a scrape, we're covering it with band-aids and casts and things that have certain mechanical properties, things that have certain light blocking properties. What do you
Hmm.
think about when it comes to the sort of the traditional things that we use in modern society to cover up or try and restore our broken bodies? Is there something here that might be that might tie in? Is there something we might be upsetting by, say, blocking light from getting to a broken bone or to a cut or something like that?
That's a good question. I'm not sure we're kind of inhibiting anything. I think evolutionarily we've kind of evolved to kind of be the best versions of these processes as possible, but I think we can make them better, which we are.
Mhm.
Post-surgical light stimulation is now being utilized, especially within the red spectrum,
Hmm.
to accelerate wound healing. Even starting to understand that at post-surgical scars to keep them hydrated. The fact that water is extremely important, and water loss inhibits wound healing. Again, that kind of brings us to this logic. You have an injury, you lose water. We know that water is also able to interact with red wavelengths of light. So is it the fact that we keep water in our body to help absorb light?
Hmm.
So there's these logic gaps that we're starting to try to fill in to see how the physical dimension has been interacting with the molecular. And another important point that I want to point out, since we're talking about wound healing and light, whenever you have an injury, you do have reactive oxygen species that are
Mhm.
getting emitted, which is what we are saying is the source of our light. That reactive oxygen species is crucial to recruiting your immune system to that wound site.
Yeah.
So it's basically a light beacon, that a
Hmm.
light emitting beacon that's recruiting these immune cells to help form a scar so you don't bleed out or have an infection. So there is an intrinsic that we now know is a light communication mechanism in the form of reactive oxygen species to recruit immune cells. So there's already a dimension that we understand.
Mhm.
Now we need to kind of probe further into how and why.
Yeah. I'm sorry. So are you saying it's actually been demonstrated that one of the ways the immune cells get recruited to a site of injury is they are following the light?
Perhaps, yeah. If we're saying that reactive oxygen species is a source of light, when you have one ROS being emitted, one photon gets emitted or some source, then yes, that is true. And we
But
see this also within in red light stimulation as well. So we probe red light into a wound site, you get a high recruitment of neutrophils and immune cells into that area.
okay, I see. So just the presence of light of the white right wavelength can help you recruit more immune cells to the site of injury.
Correct.
Hmm.
Correct.
Interesting. So this would imply that if I've got a cut or a scrape or something and I'm inside all day versus outside, like the light environment could play a role in how quickly or exactly how my wound heals.
Wound heals, correct? Absolutely. And again, this is a such a fascinating and multi-dimensional conversation because there's many different levels to this, right? You
Hmm.
have the physical side of, compressing a wound, the fact that you hold a wound to accelerate it. You have the optical side with the lights, and then you have the chemical side of actually repairing the pattern tissue of what happens. Um, Very exciting. Lots of work that needs to be done to really flesh this out.
Yeah, yeah, yeah. To flesh it out. Yeah. um, I think you do some work in cancer as well, and there's the whole world that could be discussed here. But just give us a basic idea. Are you working on cancer at all? And why is someone like a biophysicist who's been talking about all of this stuff that we've discussed so far? What is interesting about cancer to you from a biophysical perspective?
I am actively working on cancer, and it's these model systems and these processes, so neuroscience, cancer, regenerative medicine, all of them are a platform for me to try to understand the physics of life.
Mm.
Some of them give me a different direction and dimension of how to interact with it. So, like neuroscience, I can kind of play on the electrical side. And regenerative medicine, we can play on the mechanical side. With cancer, we can play with the photonic side and the energetic side of physics. And this is kind of where my PhD grew from, which where I saw that magnetic fields can alter cancer physiology. And I kind of flipped that question. If cancer can be interacted with magnetic fields, what is it emitting? And can we use that as any kind of diagnostic? So that's a paper that we published to show very specifically that launched this entire biophysics world for me to show that when you stress cancer cells, when you change their energetic landscape, they have a optical fingerprint. They have a light fingerprint that we can differentiate between healthy tissues. So we've shown this in cancer cells in dishes. We've done this in mice that we've injected with different kinds of cancers and used it as a platform, used these light signals to say, is there cancer in the animal or not? And what we're hoping to do is in humans, and that's what we're working on right now, is use these optical sensors to see if we can classify whether a patient has cancer or how far the cancer is in terms of its growth, non-invasively from the outside of their body.
Wow. So if I'm hearing you right, what you're basically saying is, okay, obviously cancer cells are different than non-cancer cells. They became different. They have a different type of metabolism. And to the extent that, we said that these biophotons get produced from based on metabolism that's happening, the cancer cells are metabolically distinct from the non-cancer cells, and therefore you would expect them to have a different photonic signature that they emit. And what you're saying, I think, is that you could basically detect cancer cells because they have this unique light signature they're emitting, and you can do this in a fairly non-invasive way.
Absolutely. That's exactly what we're doing. It's taken a lot of data points, obviously, because there's heterogeneity between cancer that we're starting to see. I mean, starting to see it's well established now, but yes, you can tell the difference between what is cancerous and what is not cancerous. What we're having a hard time doing right now is telling the difference between the cancers. If we're
Mm-hmm,
looking at
mm-hmm.
melanoma versus breast versus glioblastoma, what is that optical signature? So this is where we're looking at more of the physical dimension of the light, so the wavelength, the intensity, the frequency patterns, what is different about them? And this is where we're kind of leveraging AI to help us figure out what those pattern differences are.
Mm-hmm, mm-hmm. And I think going back quite a long time, people have had ideas about what cancer is and how it arises. Obviously, one of the classic textbook ways people think about this is what people call the somatic mutation theory. You get certain mutations and oncogenes, and maybe that causes cancer. I've had other people on the podcast that have a very different view. People like Thomas Seyfried, who say, "Well, no, this is cancer is mostly a metabolic dysfunction thing, and it's not so much a somatic mutation thing." And obviously, when we start thinking about things like light, um, people always associate UV light as causing mutations that causes cancer. How do you,
Mhm.
as a biophysicist, think about what cancer is and how it maybe ties into concepts from the regeneration field? Do you view this as primarily a
Mhm.
mutation thing, as a metabolism thing? How do you start to wrap your head around it?
Absolutely. And I'm actually kind of working on a paper on this to help solidify this idea from an energetics point of view. And I view it as a dysfunction in energy in terms of how the energy is regulated and how the energy is dissipated. And the mutations that arise might become because of shifts in wavelengths and the energy metabolism of the cell itself. We know that from our papers that there's a shift in wavelength from blue to red, and you can kind of fingerprint those. And the more you lean towards the blue, you emit more UV photons. And if those UV photons lead to more mutations, then that accumulation will change the energy dynamics within the cell. And then the cell will need to adapt. So that's where the free energy principle comes in. The cells will always do the behavior that it requires the least amount of energy for the system. So there's a different multi-layered dimension here is that I believe that it's fundamentally energetics. When there is changes in energetics, the physics match that to produce a behavioral response that the cell can adapt to, and which is what we see as highly proliferative and highly plastic. And you brought an excellent point there about plasticity, about stem cells and cancer. Why both of them are highly proliferative. One is very plastic, but the other one is also plastic, but very mispatterned. Where's that information loss
Yeah.
that allows something to become patterned versus not? And I think that's really what I want to get into is information. They're both energetics. They're both functioning at, physical and chemical landscape, but where's the mispatterning happening?
Mhm. Say a little bit more about, so what did you say around cancer cells, being more or less red versus blue shifted? What exactly did you mean there?
So the photon emissions that are being emitted
Yeah,
from cancer cells are more blue shifted than their healthy counterparts.
I
So
see.
we've seen this within melanocytes and melanoma, is where we've spent most of our time on. The melanoma cells and the tumors that are forming within rodents were more blue shifted than they were red shifted than their melanocyte counterparts.
Okay, so if I'm putting some of those ideas together, some kind of energetic disturbance happens and a cancer cell forms some way, somehow. And it's now energetically different than it was before it became a cancer cell. There's this free energy principle that I think you were pointing to. And basically, what you're saying is cells are generally going to try and be energetically efficient and use no more energy than they probably want to or need to. And so maybe in some sense, a cancer cell is one in which the cell is adapting to an energetic shift to obey this free energy
Mhm.
principle, but it's disturbed in a way that makes the cell's adaptation at its own level counter to the organism's.
Correct.
Yes. Okay.
Exactly. So the decisions that it makes, it anthropomizing it,
Yeah.
is independent of the collective of where it came from. And
Yeah.
so that disconnect is where we get, I think, tumors. But ultimately, it's arising from this change in energetic states, whether that be because of photonic inputs or dysregulation in photonic inputs. For example, we know that flavins and porphyrins, which are molecules that absorb red light, are altered within cancer.
Yeah.
So, is that the cause? Is that the result? Those are the gaps that we need to fill.
Yeah. Yeah. And yeah, lots of question marks, lots of exciting questions to pursue here. But yeah, obviously the role of light in biology extends way beyond image formation and way beyond even just circadian regulation. This stuff is fascinating. Do you think, how much has this caused you to think about the modern light environment? So, just from first principles,
Mm,
and you think about it from an evolutionary perspective, clearly our light environment that we're inside of today is very, very different from the one our ancestors and pretty much all organisms evolved in. And so,
mm.
the extent to which a lot of the stuff we've talked about is true to any degree would imply that the change in the light environment itself is probably having potentially sizable effects on our health and our biology and how it's all working together. At a high level, how do you think about what the modern light environment is doing to people?
I think about this all the time. I think about this all the time because I think about even just about 150 years ago, 100 years ago, 50 years ago, our parents and grandparents, in terms of being around natural light, candlelight, the intensity, the warmth of glow. I mean, you can go down to your neighborhood construction store and you can find cool light versus warm light. So we have this population of people who are in different colors of light, intensities of light. Um, I think about this all the time because I wonder what it does to behavior. If we
Mhm.
scale it up from molecular all the way up to behavior, I wonder what it does. And I don't have an answer in terms of what it could do, what it means for us, but this is a really cool thing to think about and to investigate perhaps in the future.
So we've covered a lot of ground, and you sort of, you've got a unifying thing that you're interested in here. But as you mentioned, you touch a lot of different aspects of biology that tie into this, from the neuroscience to the regeneration to the cancer and so forth. What are some of the things, very quick in the last few minutes here, that your lab is working on, or that other labs in this general realm are working on? Areas where you think we're going to make progress and start to learn some interesting new stuff about the biophysics of life in the next, say, 3 or 4 or 5 years?
Oh, I love it. I love that we're ending on this. I think the biggest movement that we're going to make is stepping outside of the biomolecular domain and accepting that there are other concepts that might drive and pull the genetics and the molecular. The other aspect is the energetics domain is, in my view, one unifying fundamental theme. It's a fundamental in physics. It must be a fundamental in other domains that drive biology. So, the biophysical community has been kind of also segmented, so and independent of biology. In a lot of cases, mainly because of the tools, and we kind of mentioned this a little bit. So, the next 3 to 5 years, I see a big movement, and thanks to biomedical engineering, when people are actually seeing that stiffness and conductivity and photonic aspects of the materials guide biology. And I think that will help us deepen our understanding of, if we understand that these physical landscapes alter the biology, then these physical stimuli are also in our environment, must
Yeah.
be shaping biology as well.
Yeah. So it sounds like th- you probably think, and correct me if I'm wrong, that the raw physical composition of our environment, from the physical material that touches us to the light and electromagnetic environments we're embedded in, this is probably having a systematic and perhaps underappreciated role, function in terms of how life unfolds at the behavioral level, at the developmental level, at the regeneration and disease level, than maybe biology has appreciated until recently.
Yep, that's exactly it. And if we're thinking, 20, 30, 40 years in the future, when we're thinking about space flight, and there's a domain of individuals who are looking at, colonizing other planets and things like this, we really need to understand this physical landscape for the body and the structure that we have evolved in our current physical space, if you are thinking about extending beyond it.
Wow. Well, fascinating stuff. I'm definitely going to sort of bookmark you and watch what this field is coming up with in the next few years. Any final thoughts you want to leave people with that have to do with your work or anything that we discussed today?
I think I'm clearly passionate about trying to understand the physical dimensions of life. I think as we move forward in this space and as we gain momentum, I think thinking big picture and thinking also first principles of that guide life would be something that I would kind of want to put out there and for people to appreciate.
All right. Well, thank you very much for your time. Fascinating stuff. And I look forward to talking to you again in a couple of years.
Awesome. Thanks so much, Nick.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.